The overall goal of the following methodology is to visualize and quantify the locomotion of Drosophila melanogaster larvae, allowing high-throughput screening in free-moving conditions. This is achieved by creating a tracking surface on the FIM setup and using frustrated total internal reflection for indirect illumination. Larvae of different genotypes are then placed on the surface, and crawling behavior in response to a heat gradient is recorded. The videos are then analyzed by the FIMtrack software, and data obtained are used to generate plots describing differences in larval locomotion patterns. The results show that precise, high-throughput, flexible, and easy-to-use quantifications of larval locomotion can be done based on the FIM imaging technique and the associated FIMtrack program.
The main advantage of this imaging technique over existing methods, like using incident or transmitted light, is that only animals touching the surface are visible, resulting in a superb foreground-background contrast with no disturbing artifacts.
Though this method can provide insight into biology of Drosophila larvae, it can also be applied to study walking behavior of flies and locomotion of other model organisms, such as C. elegans, planaris, or even root growth of plants.
Visual demonstration of this method is critical to convince others that a complex technology is easy to use.
The first step is to create a moist crawling surface for the FITR-based imaging method, or FIM. To do this, boil 0.8% food-grade agar in deionized ultra-pure water.